absolutely delighted now to introduce again Dr. Rob Coleman, who is going to talk to us today about recent discoveries in gynecological cancer clinical trials.
Thank you. It's great to be back here. We're just popping between three rooms here, just back and forth, back and forth, back and forth, but I'm here now. Um, so this talk was actually really hard to put together, which is a good sign because I'm trying to put together kind of the recent wins that we've seen with across the GYN spectrum of diseases. And really only had time to cover kind of the three major ones, but there's a lot going on. So it's great to see this investment of time and effort really starting to pay off. And so I'm going to review some of those. These are my disclosures. So I thought I'd kind of start with cervical cancer first and walk through a little bit of the progress we've made there, and then uterine, and then finish up with ovarian cancer. And we'll talk a little bit about kind of capitalize on what you just heard in the last talk. So let me start with cervix cancer. So, you know, the distribution of early disease and late stage disease for this disease varies all over the world. So depends on what country you're coming from, the resources are allocated, the development of screening programs, vaccination programs, all those affect these proportions. This is what we see in the United States. About half of the patients are when they're diagnosed with cancer. So many more are diagnosed pre-invasively, which is something we can deal with. But there's about half of them are early stage and half are late stage. And a lot of the progress that we've seen of recent has been in this kind of latter box, patients who present with either locally advanced disease or have recurrent and metastatic disease. And so, there we go. So one of the trials that was presented earlier this year, about a year ago, was this interlace trial. This was a very interesting idea. The idea here was to figure out a way to potentially improve outcomes, and then outcomes here are related to progression-free survival, which is what PFS stands for, and OS, which is overall survival. And the idea here was to give chemotherapy for a few cycles, as an induction before we actually started definitive chemoradiation. And what was interesting about this trial is that it was, there's the medications used here are pretty available around the world. And so this seemed to be a single kind of intervention. And you can see that with these remarkable effects, or I guess I'm getting confused here you have the presenter's slide up here, so I can't see what the current slide is. But let me share you. This is what I meant. So these are the outcomes from this particular trial, which shows these gaps. So I don't know how many people actually understand what a survival curve looks like. In fact, I have a whole talk just on how to read one of these things. But basically what they are is that the y-axis is the proportion of patients that haven't hit whatever the event is. So on the left-hand side, it's progression-free survival, so those are progressions. And on the right-hand side is the overall survival. And what the curves represent on the x-axis is time. And so over time, as patients experience an event, which would be a progression or a death, the number of patients that are available to evaluate shrinks. And so the denominator kind of goes down as the numerator goes up. And so as time goes on, you see that there's this proportion. Now, what we like to see is that the lines are flat at 100%. That would be awesome. Unfortunately, it doesn't happen that way. What you can see here is that early on, there's a relatively rapid decline to about 35% or so of patients in both groups who have experienced a progression event, and then it kind of flattens out. And when I see something like this, it makes me think that we need to go back and figure out who those people are in the tail and who are the people in the beginning part, because we're not hitting something in the beginning part, and we're identifying a group of patients that are potentially curable, haven't ever experienced a progression. So this curve morphology, as we call that, is something in clinical research we look for extensively and try to help hone who are the patients we should be appropriately treating. Because right now, we kind of treat everybody. And that's what this was, a randomized trial where everybody got either the induction chemotherapy or they got standard of care. And then that's what you saw the difference here. So there's a lot of nuances to this trial I'm not going to really get into. There's more work to be done here. And I'm going to talk about one of the other trials that was recently presented that brings in another element of interest about what this patient population is, and that's those particular treatments that are directed towards this vector, which is HPV. So as you know, human papillomavirus is found in almost 100% of cervical cancers. And because of this, once we recognized how this process worked, and we had the discovery of the immune checkpoint inhibitors, we rapidly applied this because the immune system regulates our response to infection, and this is an infection. So we started doing these studies in patients with recurrent disease who had had measurable tumor, so something we could monitor. And we gave them these immune checkpoint inhibitors, and these are just the ones that I've listed here. There's been many, many more of these now. But they all basically demonstrated that there was a proportion of patients who appeared to benefit from this. And of course, once we identified this, the development was very rapid. And with respect to chemotherapy, which I'm going to share with you in a second here, but what we ultimately wanted to do was to figure out a way to give this to newly diagnosed patients. And this trial that have appeared at Keynote A18 was addressing that particular question. So it was looking at using this immune checkpoint inhibitor called pembrolizumab in combination with chemotherapy versus chemotherapy or chemoradiation alone. And there was this maintenance phase. So after they finished the chemoradiation, then it was given for a duration of time afterwards to see if we could prevent that. And so now that you guys are all experts on how to read survival curves, you can recognize that this basically shows this difference in progression-free and now in overall survival. And these are new results. They were just updated at the meeting just a couple weeks ago with respect to overall survival. So again, the curves are relatively flat, and this is going to be in stark contrast what I'm going to share with you in the recurrent setting. So, but this represents, I think, a massive change in care. Now, one of the issues we suffer with cervical cancer is about availability, right? So drug account availability for an expensive medication like pembrolizumab is not something we can see globally. And so if you contrast that with the chemotherapy option I just shared with you with Intralase, there's this opportunity that we have a kind of a broad spectrum of opportunity. What we really need is prevention, as you heard in the last lecture. We need prevention because that would ultimately normalize this burden of healthcare around the globe. We also saw in this trial that there was an increase in objective response and also in duration of response. So again, kind of a triple win. We were able to, the patients who had measurable disease, we were able to make those tumors shrink. They were able to stay away from progression longer, and they lived longer. So that was a big win. When when we first started moving immune therapy into cervical cancer, as I mentioned, we did these single-arm trials where all the patients enrolled in the study had various different prior exposures to therapy, and then we were given immune checkpoint inhibitor to see if they responded or not. When we saw that signal, the first thing we did was to think of how we could add this to chemotherapy. And this was done in two trials. One was recently reported, one reported last year, to our best kind of chemotherapy backbone. So in cervical cancer, we've done a lot of work in the past to add bevacizumab, which is a drug targeting angiogenesis, the development of new blood vessels, to chemotherapy. And we showed that we were able to benefit patients with that triplet. So what we decided to do was a quadruplet. And so there were two trials. One is called 826, which, as you can see, is pembrolizumab. I have a laser here. Pembrolizumab, which is the immune checkpoint inhibitor given to our backbone of chemotherapy, and you can see was an investigator choice plus minus chemo. And in the BEAT-CC trial, which was very similar, again, it was paclitaxel, carboplatin, and bevacizumab was required in everybody. So they're slightly different, but they came home with the same message, and that was that for progression-free survival and overall survival, there was a substantial win. And we knew about this last year, and then of course now we have a confirmatory trial with basically the same design, which shows that there's an improvement in progression-free survival and in overall survival. So in the United States, the pembrolizumab is already available for treatment for this group of patients, and we'll wait to see if the atezolizumab actually also makes it in. Two different types of immune checkpoint inhibitors, but basically targeting the same axis. And we also showed, again, objective response improved and the duration of response. This duration of response metric is now really important from the standpoint of the patient experience, because what we're trying to show is that when the tumors are shrinking, we kind of start the clock there and we monitor how long that benefit is obtained. We usually combine that with the quality of life PROs to determine whether or not the duration of time while on therapy is actually balanced against the toxicity of being on for a longer period of time. Because if you're responding, you're going to stay on treatment. And so the idea is now is to try to figure out ways mathematically to separate out the toxicity from the benefit so that we can get a global overall picture of what the patient experiences. And that work is continuing to evolve as we get better and better tools to understand what is the patient experience. So to put this into some kind of context, I've listed this, I made this slide a couple years ago and I kept adding to it. During a lot of my professional research time, we did a lot of these trials here, we kept adding different drugs, and we basically, in terms of overall survival, had kind of a flat curve. And then we started with this addition of bevacizumab, as I mentioned, and that saw us improvement. And then ultimately, when we got to the addition of the immune checkpoint inhibitors, we actually increased from what was our best standard about 150% on overall survival expectations. And what's really remarkable, if you go back to the old days, it's almost a four-fold increase in expected survivorship. So that is the work. Like I said, a lot of hard work being done there, but for years we didn't make any progress. So we needed something new that attacked the biology, and that was where the real success was here. Kind of the next wave of these, and you'll see this again in the endometrial, and you'll see it in the ovarian cancer, is this idea of an antibody drug conjugate, and it's just what it says there. It's an antibody that targets a specific antigen, and it's linked to a chemotherapy that you can't really give by IV because it's too toxic. And so it's kind of a smart bomb or a kind of a directed chemotherapy to the tumor. And so the first of these compounds that we evaluated that I was fortunate enough to be involved in was this tisotumab vedotin, which targets tissue factor. And this was a randomized trial that was recently reported that showed this combination, or I mean this single agent against investigator choice chemotherapy. And what we saw here for the two major endpoints is an improvement in both progression-free and overall survival. So again, now you'll notice these curves. Remember I said they would have a different morphology. So this is a group of patients who have been through the front line and been through the combination second line, and now are getting this as a third line drug. And you can see it's much more difficult to treat the further we are, but we're still making progress in each one of those settings. And as I mentioned, the good news here is that now that we've seen that the index case works, is now there's been an explosion of these drugs coming. And so these are just some of the different targets that are out there, one of which has been recently reported, and that's targeting HER2, which is something you may have heard of from the breast cancer world. We have this drug called trastuzumab deruxtecan, which is approved in GYN cancers for a 3+ expression, but essentially it's the same thing. It's the antibody targeting the HER2, and it's bringing the chemotherapy, which in this case is a drug that has a specific target called topoisomerase 1. And you can see these waterfall plots, which represent the depth of the responses that we saw in this experience. So this again provides us some really interesting opportunities as we go forward. So I think in cervix cancer, disease that we really had very hard time making any progress in the last 10 years, we've really made significant improvements in life expectancy. New drugs are now entering the market as we understand more about what the immune response is to this infection. And we're capitalizing on the expression of various different targets with these new molecules. So lots of exciting new work happening here. Switching gears a little bit to endometrial cancer, this is another disease for, gosh, most of my professional life had a very difficult time getting anybody interested in wanting to study something new. I think most people felt that you had radiation, you had surgery, and you had chemotherapy, and that was good enough. We knew that there was potentially a hormonal axis because some of the endometrial cancers that we see have this relationship to hormone receptor expression and response. Known that for years. We really didn't understand what was going on with endometrial cancer and the potential opportunities until we started getting molecular data. And what you're seeing here is a very famous publication from what we call the Cancer Genome Atlas, or TCGA, which is now profiled over almost 40 different tumors this way. They did extensive genomic and RNA and protein evaluation. And what we learned in this particular experiment was that endometrial cancer was a collection of very different diseases. Some of it looked just like breast cancer, some of it looked just like ovarian cancer, and some of it had this relationship to something we see in colon cancer, which you just heard about in the last talk, about these microsatellite instability. And then there's this fourth category, which we call no specific mutation profile, which actually has a number of categories within it as well. So once we learned that this is actually a spectrum of diseases with unique targets, combined with all these new therapies that were coming up, we started to see a lot of acceleration in change. And one of the first things that we saw was this idea that this microsatellite instability could be targeted, just like it has been in other tumor types where we see this, like colon cancer, with immune checkpoint inhibitors again. And so our initial experience here with some of these drugs basically showed that in this in the tumor types that were deficient in those mismatch repair proteins, that they were very sensitive to a single agent of an immune checkpoint inhibitor. And because of this experience, we rapidly started to move this forward into the clinical experience in the phase 3 setting in patients with earlier presenting diseases. And these are just some of the trials that have been done and some of the results. And what I wanted to share with you is this particular graphic, because when we showed this, the first two, the GY018 and Ruby data at the SGO meeting a couple years ago, it left the room with a standing ovation because we'd never seen this kind of change in endometrial cancer. Really remarkable. We married a molecular target with a drug that hits the molecular target. And this is what we got, these big gaps. So, it's hard to see exactly what the numbers are, but all you need to appreciate is what the gap is between the two lines of those curves, because those represent the patients who got the drug and the patients who didn't. And so this was, again, rapidly, it was approved and it was repeated in many of these trials. We thought, well, maybe if it works so well in the deficient patients, the deficient tumors, maybe it would work in the proficient tumors. So these are patients that actually have tumors that are proficient in that mismatch repair process. And you can see the experience is much less robust. In fact, we only have one trial, G1018, which actually shows a difference statistically. The rest of these are basically overlapping. So what this tells us is that in this other subgroup of patients, we now have another kind of investigation we need to do to see if there's another subgroup in here that we might be able to actually benefit. One of the thoughts was to combine a PARP inhibitor, which we use in ovarian cancer, to this patient population because it shared a number of those characteristics. Unfortunately, the two trials that I've listed here make it very difficult to conclusively determine whether or not the PARP inhibitor added to this backbone that I just mentioned is actually working. So these are some of the results that we saw from the two trials, DUO-E and Ruby Part 2, that have tried to address this question. I think we're still trying to kind of sort it out. With more work and more time, hopefully we'll be able to identify the appropriate group that needs to get this therapy. One thing I will say parenthetically is that if the patient's tumor has that deficient MMR status, they don't really need the additional therapy. We're getting most all of the benefit out of that immune checkpoint inhibitor in combination with chemo. So just recently, just a couple weeks ago, we saw, and it's going to be presented here at this meeting, is an update on this B21. So this is actually moving this exciting new therapy that I just shared with you in the recurrent and advanced metastatic setting to an earlier line of therapy. And so you can see over here, it's the same chemotherapy plus or minus pembrolizumab, followed by a maintenance therapy in a patient population that is much less heavily pretreated. So we were very excited about this trial, but its trial design was basically looking at the intent to treat population, so everybody. Now, what you've just seen so far is that there's this differential between the MMR, the deficient MMR population and the proficient MMR population. Here they're just combined. So when we got the initial report of this last summer or this summer, that there was no difference between these arms, we were very, I guess, we were upset to see that this was the case. But what you're going to see here, actually, I don't know if I can share it. Actually, oh, I can't share it. Let me just say, I'll hint and say that, listen, what you saw before is going to apply to this, but you didn't see it from me, you heard it from her. So yeah, unfortunately, I can't share it with you, even though I just shared it with you. Okay, so, but the good news is that, again, the biomarker is coming through, and you'll hear this over and over again, that these biomarker identifying specific tumor vulnerabilities against a drug that works against that vulnerability is actually leading to benefit. And so because we've seen this exciting result, the one big thought is maybe we can get rid of chemotherapy altogether in this patient population that carries the tumors with this alteration. And so there are a couple trials out there that are left to report. We have one that has reported that essentially is negative, and this is the, oops, let me go back. And this is the data. So you can see the curves basically overlap. So we don't really have a strong signal here. As I mentioned, there's still 2 more trials ongoing to see whether if we can get rid of the chemotherapy altogether. And then I know I'm running out of time here, but let me just talk a little bit quickly about the ADCs again. This is that antibody drug conjugate I just mentioned to you. In cervical cancer, we see the same thing here in endometrial cancer, again, seeing a benefit and with objective responses on these waterfalls. And fortunately, now we have 2 drugs. So that's one, and this is another. And just to highlight, and these waterfall plots, they take a little time to explain to you, but what the length of the fall down going down is the magnitude of the response to the tumor shrinking. So if it goes all the way away, 100%, then it goes all the way to the bottom. And then everything else that's below that dotted line is considered a partial response. And that's how we kind of report these data. As I mentioned, there's so much going on here. There's so many more drugs under development. It'd take way too long to explain it, but the good news is that there are a number of these trials which are actively accruing patients. So I mentioned that there's a cohort of patients whose tumors also carry a vulnerability to endocrine therapy. Just like we see in breast cancer, there are a number of targets that we've been studying over the years. We've known about this. Right now, there is one trial that's launching that's actually affecting 2 different components of this endocrine axis, which we also see in breast cancer, and it's getting ready to launch. So we're really excited to see this going forward. And the last piece I wanted to mention is something that I'm also very passionate about, because it's a trial that I'm responsible for, is this looking at this other cohort of patients, as I mentioned, that there might be in this group that carry a proficient MMR status, but have a wild type for this P53 gene. So most P53 gene is one of the most commonly mutated or altered genes in all of cancer. And almost over 95% of ovarian cancer has a mutation in P53. There is a cohort of endometrial cancer patients that have it intact. And what this new drug, which is not new to the hematology world, but it's new to the solid tumor world, selinexor, is actually trying to keep the normal functioning protein from getting excreted out of the nucleus into the cytoplasm where it gets degraded. So this is a selective inhibitor for nuclear export. And so we had a strong signal that in these patients who got this drug who carried the P53 normal, so non-mutated P53, that there's a huge benefit, which we see over here. And so that led to an ongoing trial now called Export-EC, which is trying to prove that in patients that carry that signature. So again, molecular biomarker, drug targeted to the biomarker. So you want me to close here, or you want, because I know you got more.
We can do.
3 minutes?
Yeah.
Okay. All right, I'll run through ovarian cancer. There's not as much going on, surprisingly, in this disease. But one of the things that has been the staple of our most recent advancements has been this combination of angiogenesis and PARP inhibition, which you heard in the last. And they've been applied at all of these yellow points as opportunities to improve and maintain the molecular or the biological suppression of the cancer growth process. So these 2 drugs have been extensively worked up, and they have now entered into the guidelines particularly in the primary setting, where I've outlined here where bevacizumab is being utilized as part of the initial therapy. And then there is this decision-making process based on the genomic signature of the tumor, which you just heard about in the last talk. And then based on that, what kind of therapies are now recommended. So what we're trying to do now is we're trying to figure out ways to add to this, because we've demonstrated some pretty strong treatment effects. And so one of those is to see if we can add immunotherapy to this kind of doublet opportunity. And one of the trials that was recently reported is called the DUO-O trial, which is looking at the combination of olaparib along with durvalumab, which is an immune checkpoint inhibitor, with bevacizumab. So there's the 3 kind of drugs. And it basically shows that that in the patients that carry this genomic signature, the HRD test positive tumors, that there's a decent treatment effect here with the triplet. But there's already a strong treatment effect with the PARP inhibitor alone. So it's been very difficult in this particular trial to kind of sort out exactly what's actually contributing to the benefit in these two or three different populations. So fortunately, with time, we'll have a better clarity of this because we have at least two more trials that are ready to report. And so hopefully next year, we'll be able to talk a little bit more about where we think this triplet kind of strategy is going to go forward. So to capitalize on what I just mentioned with respect to these antibody drug conjugates, the third kind of component of what's being evaluated now in recurrent, platinum sensitive, and platinum and in frontline disease is the ADCs, the antibody drug conjugates again. And just to give you a flavor of what's going on here, there's just so many of these drugs again that are being applied across the board because they have this efficacy signature with the expression. We have not had 100% wins. We evaluated three different strategies here. One was using an adenovirus vector to try to deliver a gene. We had an inhibitor for Gas6-Axel as a decoy receptor, and we had this idea of tumor treating fields. And we compared each one of these novel therapies in a +1 design. So everybody got paclitaxel, and then it was either half of the patients got a +1, so one of the new agents. And unfortunately, we did not succeed in any of those events. So, we hope that with better biomarker development and preclinical work, we can actually come up with the better strategies to not have to have this problem. But that's unfortunately part of what happens in the clinical research experiment is to try to figure out which of these agents and what kinds of things can be added to be beneficial. But not all hope is lost. The first ADC that we were able to evaluate was mirvetuximab, and it showed a benefit in both progression-free and overall survival. The first time we had ever shown that in both of those endpoints. So we were very excited about that. And because of that success, we've seen a rapid evolution of the ADCs, and they're coming. So hopefully next year we should have some more results with earlier lines of therapy, including the maintenance strategy. So to kind of summarize this, I want to kind of keep the eye on the prize. We're trying to continue to make progress to move each of these selective endpoints, if they're even the intermediate and early endpoints going forward. I love this motto. I've used this since like 1996, that research cures cancer. It's my kind of like I stand behind that staple because we never make any of this progress unless we actually conduct these trials. So I appreciate all of you who have contributed to the process of doing informative clinical trials. And I'll go ahead and close with that. This is Ireland on the West Coast.
Thank you very much, Rob. Any questions? It was an amazing whistle-stop tour of all the research.
Yeah, there's so much to talk about. Yeah,
it's quite overwhelming in some ways.
Mm-hmm.
Has anyone got?
Yes, ma'am. Mm-hmm.
Just to make sure that the patients are first and foremost thought about, and we kind of between us we read the trials and we discuss the implications and how it impacts the patients as well. And we have, well, they've added a slide to kind of show that as well. Do you guys do anything like that?
In adding the patients to the
Adding a patient's perspective when you're presenting.
Yeah,
clinical trials.
actually, we have a session coming up on Thursday where we have a patient who's going to be at the table to talk about this. Yeah, no, we're very much aligned with that. And actually, all these trials have patient advocates that are on the clinical trial development team and help us with the experience. Because a lot of times when we write out a schema, we might say, okay, this is the perfect schema. We'll be able to answer all these great questions. And then we'll hand it to somebody like Annie Ellis, who was going to be here, and she'll like, that you're not going to be able to do that. Nobody's going to come back for that, or it doesn't make sense to do that. So we rely heavily on the advocacy community to not only develop better trials, but also for the grants. It's so critical. Hmm. Yes, ma'am.
Lovely presentation, and it's great to see all the progress. Just in relation to all the ADCs that are coming down the line, how is the health system going to cope with all of those? I mean, they can't all be reimbursed. So what do you think the difference is? What are they going to look for between the ADCs?
I love it. Oh my gosh. Yeah, this is something I've been, me and several people have been very passionate about. Right? I think that the kind of the shotgun approach, the Me Too's that are developing out there, we're going to have to start comparing these against each other to find out which one's actually the best. And fortunately, there has been some work done that way in breast cancer with respect to the HER2 axis. Well, the antibodies looked good, and then we went to the first ADC, and that looked good, and then we actually compared it to a different ADC, and it was better. And so now we've been able to isolate those subgroups into where those groups work. What I didn't talk about a lot here is that each of these ADCs kind of target a specific antigen that's on the surface of the cancer cell. And there's sometimes a lot of antigen and sometimes there's a little bit of antigen. And we can actually capture that on some of the stains that we do with the histology. And so many of these ADCs work really well in high expressing antigen loads. But if they're only 1+, they don't really work all that well. And so maybe there's a different ADC that should be used in that particular setting that doesn't have a dependency on the expression. So that's the kind of work that needs to happen next. And so yeah, we're really amping up the heat on to make companies do that.
Thanks so much for your talk. I had a question about the Duo study that you mentioned, where you're looking at bevacizumab, the anti-angiogenic. I saw in your survival curve that you were looking at the HRD positive tumors.
Mhm.
Are you looking at high-grade serous
Mhm.
ovarian cancer for that? Is there any other kind of stratification by transcriptomic subtypes? Because there's been a bit of evidence to show that based on, the types of genes that they're expressing, they might actually respond better to bevacizumab. Is there the infrastructure for that? Or?
There's a huge amount of interest in that. And look, I mean, bevacizumab was first developed in 1997, 96, and we've been looking for a predictive biomarker since that time period. So yes, it's incorporated in that. We don't have a predictive biomarker as yet, but there's a lot of interest, obviously, and a lot of work that I'm sure you guys have done, and some of that we've been involved in trying to identify exactly what is contributing to the lack of response. Because we don't see universal response with bevacizumab either. But the combinations are harder, right? So when you have two drugs together, you have to sort out what is the relative contribution of each. And when you have three drugs, you can do the math. It's eight different potential comparators. So it becomes very difficult, which is why I said this is going to be difficult to interpret. The HRD signature, what you didn't see up there was patients that whose tumors carry a BRCA mutation, because that was already felt to be kind of addressed appropriately by just the olaparib or just the PARP inhibitor. So for the HRD test positive, which is a heterogeneous group, we're trying to figure out better ways to identify those patients. But you're exactly right. That's the homework that we need to do. Mhm.
Yes, thank you so much.
Sure.
I'm also interested in rare gynecological cancers. So we saw that there is much going on in gyne cancer. How about the rare gyne cancer?
Yeah, this is a great question, and there are some programs now that are actually starting. And you mentioned granulosa cell and other germ cell tumors that we do have programs starting. They're harder to enroll for randomized trials. Obviously, the easiest one for us among the rare tumors is low-grade serous, and so we've done three now randomized trials in that setting. But for the non-epithelial tumors, it's been difficult. And actually, the bar, from a regulatory standpoint, we don't have to do that formal 600 patient phase 3 study. We just need to have a strong enough biomarker and a signal. And I just reviewed another application that's actually looking at the FOXL2 gene alteration, which we see in some of these rare non-epithelial tumors as a specific targeted agent for that. So it's common. It's just taking longer because we need to get people to get together to form a collective that we can study. Mm-hmm. Yes, ma'am.
One more question and then.
Okay. I understand you need to have clear outcomes like overall survival and progression-free survival, but for patients, the quality of life is of course very important. It's great that you have maybe 2 more months, but if it's a horrible 2 more months, so how is quality of life a factor in these?
Yeah, great question. I mentioned a little bit about this, that we are using some different analytical tools now that are able to subtract the benefit that you see in the PFS curves against the toxicity that's actually mounted for being on treatment. And patients that are benefiting are actually on treatment longer than the patients that are not benefiting. So it's important to be able to put some context with that. I would say that and I've had this discussion with the US FDA, that they are welcoming us to come up with some kind of metric that would be able to capture that in a prospective way as an endpoint for a study. But we haven't really come up with one validated tool yet. But these techniques, such as the Q-twist or the quality adjusted twist analysis, essentially does that. It subtracts out the toxicity from the benefit, and then it shares whether or not there's still a benefit. Because 2 months, is, essentially is not at the median is not really a clinical benefit. So what we try to do is to see that even if they have toxicity, is there still a benefit that's gained? And so that's kind of where we are right now.
Okay, thank you very
All right,
much.
thanks,
That
you
was
guys.
brilliant.